Hello everyone. Welcome to the session of Sexual Reproduction in Flowering Plants, Theory Part 5. So today we are going to study about the sexual reproduction in flowering plants, and today we will discuss about the double fertilization. Double fertilization. Disclaimer: the images, text, and other material used in our presentations are for educational purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issue, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using generative AI, which is experimental. So, this is the learning objective of today's session. So, what we are going to learn today to understand the fertilization in flowering plants involving double fertilization and triple fusion. Okay. So, we will see how the double fertilization occur in the flowering plants. First thing. Second, to understand the two male gametes produced from a single pollen grain and are necessary for the double fertilization. We will also see how the two male gametes are produced in the from the single pollen grain and how they are necessary for the double fertilization. And to understand the stage of embryo development in the monocot and dicot and endosperm development. Actually, the result of the first fertilization, that is the syngamy, is the embryo, and triple fusion, the result is endosperm. So we will discuss about the embryo and endosperm in a very short form today, but in detail we will discuss about the embryo development and the endosperm development in the coming lecture. So in today's lecture we will study about the male gamete, female gamete, as well as the double fertilization. So this is the index. We will study about the male gametophyte, though we have already studied about the male gametophyte, but for double fertilization we will see again the male gametophyte as well as the female gametophyte. Okay, and then we will discuss about the double fertilization. We will also summarize the today's lecture at the end. So, without wasting any time, let us see about the male gametophyte. Male gametophyte. This is 1.2.1 part of your NCERT. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. This is the size of the pollen grain. Pollen grain, okay? This is the size of the pollen grain. Then generally the pollen grains are of 25 to 50 micrometer, and that is measured in diameter. This is measured in the diameter. So they are telling about in flowering plants the pollen grains are spherical in shape, generally spherical in shape, and they measure, and when we measure the diameter of it, it comes in between 25 to 50 micrometer. So it has a prominent two-layered wall. We have discussed about the two-layered wall. Can anyone tell me what are the two-layered wall of the pollen grain? You can give the answer in the chat box. Two layer of the pollen grain, and that is very prominent. That is very prominent. Intine and exine, very good. Intine and exine, yes, they are having the two layer. The outer wall is called as exine, as it is present outward. Okay, and the inner one, that is intine, that is called as intine. Now, the hard outer layer called exine is made up of sporopollenin. This is the chemical chemical nature of the chemical constituent of it, that is called as sporopollenin, which is the most resistant organic material known. Now we can think about what is the organic substance through which this sporopollenin is made up of. Organic substance, I'm asking the organic substance by which this sporopollenin is made up of. Though it is not mentioned in your NCRT book, but yes, you should know what is the chemical, whether it is made up of carbohydrate, it is made up of protein, it is made up of fats. What are the things that make this sporopollenin? Hmm? So the sporopollenin is a very high resistance biopolymer. It is a biopolymer. Polymer means it must be having a one monomer and that get repeated and form a polymer structure, right? So it is a biopolymer and primarily composed of long chain of fatty acid. What it is consist of? Long chain of fatty acid. First thing, phenyl propanoid. Second thing, the chemical constituents of the sporopollenin. First thing is the long chain of fatty acid. Second, phenyl propanoids and phenols, along with small amount of carotenoids. Small amount of carotenoids. So four things are there. Again, I'm repeating. Sporopollenin is made up of or consist of long chain of fatty acid. So it is having the fatty acid, lipids. Okay. Again, it is having phenyl propanoids. The chemical name is phenyl propanoid. Second thing. Third is the phenols. Phenols. And fourth is the small amount of carotenoids, or the carotenoids are present in the traces. Traces mean in a very small quantity, right? So this is about the exine. Exine is made up of sporopollenin, and now we know what the sporopollenin is made up of, its chemical nature, and it is resistance to organic material known. It is the most resistant organic material known. What is the meaning of resistant? Resistant. It means it can withstand with the high temperature. High temperature, strong acid and alkali. Okay. If you treat it with the alkali, it will not destroy the sporopollenin. It will, if you treat it with the alkali or acid, or keep it at a very high temperature or a low temperature, there will be no effect on the sporopollenin. It is so hard substance. In fact, it is the most resistant organic material known, right? Very important point. So here you can see the structure of the microspore, or you can say a pollen grain, right? We will see. We will see. So no enzyme that degrade the sporopollenin is so far known. There is any enzyme, you cannot tell any name of an enzyme that can destroy or degrade the sporopollenin. No enzyme is known so far that can degrade this sporopollenin. That is why it is the most resistant organic material we know today. Pollen grain exine has a prominent aperture called germ pores where the sporopollenin is absent. Okay. Now this exine part, if you think about the exine part, at some particular point, there is an absence of the sporopollenin, or the sporopollenin is present in a very small amount. Very small amount. So that particular point are called as germ pores. Very important. Germ pores. And these pollen grains are well preserved as a fossil because of the presence of sporopollenin. Pollen grains that have been deposited in a very long years back, but it is viable. It is preserved as a fossil because of the presence of sporopollenin. So this sporopollenin is very, very important. The exine exhibits a fascinating array of the pollens patterns and design. Now, the inner one, the inner wall is called as intine. Now, here they are talking about this exine exhibit a fascinating array of patterns and design. When you see the exine structure, they will have a particular pattern or design over their wall. So, this is also one of the feature of the exine. Now, these points are why so important. Why I have given all the statement in a bullet form. Can you think about why it is? Because there are so many multiple statement questions. And some of the questions come from this NCRT only. They take the one line from the NCRT and make a statement, and they can give the fourth statement, right? So that is why it is very important to emphasize each and every line of the NCRT book. So that is why each line is individual, and we are discussing each and every line here. So the exine exhibit a fascinating array of patterns and design. You must remember this line, because in the examination, sometime in the multiple statement question, they can give like the intine exhibit a fascinating array of the patterns and design, right? It comes like that only in the multiple statement questions, right? So you must remember the exine is the outer wall of the pollen grain that exhibit a pattern and design, not the intine. Okay. The inner wall of the pollen grain is called as intine. The inner wall of the pollen grain is called as intine. And what material it is made up of? Can anyone can tell me what material it is made up of? Because we have earlier also we have discussed this point. Only cellulose, Govind? Only cellulose or something else also? Harshita also says cellulose. There is one more substance. There is one. Yes, pectin. Pectin. Yes. The inner wall of the pollen grain is also called the intine, and it is made up of cellulose and pectin. Okay. Here you can see. It is a thin and continuous layer. Now, was exine a continuous layer? Sorry. Was exine was a made up of a continuous layer? Can we say that this statement exine is a continuous layer? Yes or no, you can give in the chat box. Is exine a continuous layer? Yes or no? Here it is written about intine that it is a thin and continuous layer made up of cellulose and pectin. Okay, I got one answer. No. Correct. It is absolutely correct. Because exine is not continuous. At some point, the exine, which is made up of the sporopollenin, is absent. And that particular point makes the germ pore. That particular point makes the germ pore. So it is not a continuous layer. But your intine layer, which is made up of cellulose and pectin, it is a thin and continuous layer. It is a thin and continuous layer. And yesterday I got a very good question from one of the students. I don't know who asked that question. That pollen tube is an emergence of what? Whether it is a plasma membrane, whether it is an intine, whether it is an exine. Yes? So this pollen tube is the emergence of this particular intine layer, which we are talking about now. Yes. Okay, great. The germ pore were there at the exine layer, but the intine is continuous. The intine is continuous, and from that germ pore, the intine comes out. The intine comes out by a protuberance form, and that is called as pollen tube. That comes out in a form of protuberance, and that is called as pollen tube, right? The cytoplasm of the pollen grain is surrounded by the plasma membrane. Now, Just inner, just inner to the intine, there is a plasma membrane or a cell membrane. Okay, and they are having a good amount of cytoplasm in the pollen grain, right? So the cytoplasm is surrounded by the plasma membrane. So there is a plasma membrane, the innermost boundary. Then comes the intine, which is made up of cellulose and pectin, thin and continuous layer. Then the outermost, that is exine, made up of sporopollenin, and it is not continuous layer because at some particular point the germ pore are present. So these points are not given because it is not written anywhere in your NCRT that exine is a continuous layer. But by reading point by point, you can understand this. If it is talking about the intine layer, that it is a thin and continuous layer, was it present for the exine also? Think about that. Okay? While reading your NCRT, while discussing NCRT, think in this way. Then your preparation will be up to the mark. Okay. When the pollen grain is mature and it contains two cells, pollen grain becomes mature, it contains two cells, the vegetative cell and the generative cells. Okay. So the mature pollen grain, it contains the two cells. One is the vegetative cells and one is the generative cells. The vegetative cell is quite bigger. We have seen in the figure. It is quite bigger and it is having the abundant food reserve and a large irregularly shaped nucleus. Okay, they are having the abundant food material and irregularly shaped nucleus in it. The generative cell is small and floats in the cytoplasm of the vegetative cell. Now, very, very important point. It is so small that it can float in the cytoplasm of the vegetative cell, right? Generative cell is very small. It is shaped like boat, spindle shape. And its nucleus is again very, very small. Very small. So here you can see this was the microspore. And here you can see microspores are having the vacuoles. It is written, it is not written in your NCRT, but in some question it can be asked. Are the vacuoles are present in the this microspores? So you must be thinking that it is not given anywhere in the NCRT, but in the diagram it is clearly mentioned that they contain the vacuoles in it. They are having the nucleus. Now here, asymmetric spindle here is here. Means the spindles are formed here. It means the nucleus is divided. And when the nucleus is divided, you can see the generative cell nucleus is very small. And vegetative cell, it is quite a large cell and it is having a very prominent nucleus and good amount of cytoplasm. But when you see or compare it with the generative cell is very small and it is spindle shaped. It can float in the cytoplasm of vegetative cell and the nucleus is also very, very small. Okay. So this is about the one of the mature pollen grain, which is a mature pollen grain. It contains the two cells, the vegetative and the generative cells. So this is the mature pollen grain, one generative and one vegetative cells. This is again, you can say this is a microspore, but this is not in a mature form. This first figure, this is not a mature one, but this is the mature one. That is why it is written here. When the pollen grain is mature, it contains two cells, one vegetative and one generative cell. Now, it is the spindle shaped with the dense cytoplasm and a nucleus. The cytoplasm is dense, but a small nucleus, and the shape is a spindle. Now, this is very, very important point. In over 60% of the angiosperms, pollen grains are shed at this two-celled stage. 60% of the angiospermic plant, the pollen grains, when they are shed off from the anther, where anther becomes dry, okay, its wall get ruptured and this is shed off at two-celled stage. What is the question in the DPP? That mature male gametophyte is two-celled or a three-celled? The answer is the three-celled in the DP. Is it correct? Okay, I have to see about that. I didn't remember that like three-celled stage was there. Okay, I'll check about it, and then again we will come back to it tomorrow. I'll check about it. Okay. Okay, 60%. In 60%, it is shed at the two-celled stage. In the remaining species, the generative cells divide mitotically to give rise. Remaining species means the rest of the 40%. The rest of the 40%. The generative cells divide mitotically, give rise to the two male gametes before pollen grains are shed. It means that in the rest of the 40% of the angiospermic plant, the pollen grains are present at three-celled stage in the anther only. The generative cells divide into the two nucleus, and that is two male gamete. Okay, three-celled stage. So that is the three-celled stage. And at three-celled stage, they shed off from the anther. They are released from the anther and dispersed in the environment. Okay, so we can say that majority of the angiosperms, the pollen grains are shed at the two-celled stage. Okay. Now, in majority of the flowering plant, one of the megaspore. Now, we will discuss about the female gametophyte. Okay. 1.2.2. That is the topic in your NCERT. Now, in a majority of the flowering plant, one of the megaspore is functional with the other three degenerate. Now, I recall one of the point which I have discussed in my second lecture, that one of the nucellus cell, you understand where the nucellus is present? Think about ovule, think about nucellus, the round oval structure. It is having the funicle stalk-like structure attached with the placenta. Now, the round oval structure of the ovule and the funicle stalk-like structure attached at a particular point, that is called as hilum. Okay. Nucellus is present in the center, that is the main round oval structure. And this structure is surrounded by the involucre, or you can say the covering, which is called as integuments. Now, how this female gametophyte is formed? We will discuss about that. One of the nucellus cells become prominent, become different by accumulating large amount of cytoplasm and very large size of nucleus. It becomes very different from the other nucellus cell. And that undergo meiosis division or the reduction division. And what is the result of that reduction division? 4 megaspores are formed. And now that this particular line comes here, in a majority of the flowering plant, one of the megaspore is functional while the other three degenerate. Okay, so coming back, that 4 megaspore, out of those 4 megaspore, 3 get degenerated and only one become functional megaspore. 3 get degenerated. And what is the ploidy of this megaspore? Can anyone tell me? This one functional megaspore, what is the ploidy of it? N. Very good. N. Now, that one particular cell of the nucleus, which I have discussed just now, that one of the nucellus cell become very different from the other nucellus cell by accumulating a lot of cytoplasm and very prominent nucleus. That particular cell is called as megaspore mother cell. megaspore mother cell. What is the ploidy of the megaspore mother cell? Can anyone can tell me? What is the ploidy of megaspore mother cell? 2n. Very good. So all the nucleus cell is having ploidy 2n, and one of the nucleus cell become different, just by different because its size get increases, accumulate cytoplasm, prominent nucleus, but the ploidy is still 2n, and that cell is called as megaspore mother cell. This megaspore mother cell undergoes the reduction division, meiosis division, and form 4 megaspores. Now these 4 megaspores are haploid in nature, n. Haploid in nature, n. Okay. Now, out of these 4, 3 get degenerated and only 1 megaspore is functional. Okay, so this is the story of one functional megaspore. Always remember this. Now, only the functional megaspore develops into the female gametophyte that is called as embryo sac. The one of the megaspore is functional. So, this megaspore is involved in the formation of whole embryo sac, and that is why it is called as, Prashant told me, monosporic development. Monosporic development. Very good. Because only one of the megaspore is involved in the development of whole embryo sac. Okay, no problem. So this method of embryo sac formation, from a single megaspore is termed as monosporic development. Now you can understand why it is called as monosporic development, because only one of the megaspore is involved in the development of embryo sac. What is the number of cells in the embryo sac? Number of cell in one embryo sac. I'm just talking about cell, not the nucleus. How many cells are there in one embryo sac? Embryo sac, we have discussed about it. Anyone can tell me? No. Embryo sac. The embryo sac is formed. Female gametophyte embryo sac is having no. Okay, my question is, how many number of cells are present in embryo sac? 7 cells. Yes, 7 cells. Okay. Embryo sac is made up of 7 cells, and this 7 cells is formed from one single megaspore. One single megaspore. Right? The functional one, because 3 got degenerated. Now, how this single megaspore leads to the formation of an embryo sac, we will see that. So let us study about the formation of embryo sac in detail, right? This I was talking about, the embryo sac. So here you can see, this is the one of the megaspore. So I am drawing here one of the megaspore which is having just one nucleus, and this comes like this. So it divide by simple division, it divide. One nucleus is divided into 2 nucleus, and here you can see the 2 nucleus is there, and the 2 nucleus moves opposite pole. It moves toward the opposite pole. This we are talking about, this one of the megaspore. 3 got degenerated and 1 is left. So we are talking about how the division occur in one of the megaspore. Okay. So here you see one of the megaspore divide 2 nucleus, and after the division of nucleus there is no cytoplasmic division, or the wall layers is laid down. Okay, no cytokinesis. Sorry. In fact, after that, there is again karyokinesis. There is again karyokinesis, that is the division of nucleus. So here you can see at each pole 2 nucleus are there. 2 nucleus. In total, 4 nucleus in the this megaspore. Now here you can see it divides again third time, and it can form 8 nucleus, 4 towards each pole. 4 towards each pole. And from each pole, one of the nucleus move to the center. One nucleus from the micropylar end moves toward the center, and one from the chalazal end moves toward the center. And here in that center, you can see the 2 polar nuclei. Okay. So it can be asked whether it is coming both the nucleus, 3 mitotic division in total. Yes, 3 mitotic division. The first, the second, and the third. This is the third. So after the third, you can have 8 nucleus in 1 megaspore. 8 nucleus in 1 megaspore. 3 mitotic division. Now it can be asked in the examination that this both the polar nuclei is coming from the micropylar end or chalazal end. No, each nucleus. One of the nucleus is coming from the micropylar end, and one of the nucleus coming from the chalazal end in the center, and that is called as 2 polar nuclei. And after that, the rest 3 nucleus at the micropylar end form 1 egg cell and 2 synergid cell. 1 egg cell and 2 synergid cell at the micropylar end. And at the chalazal end, the 3 nucleus are there, and the wall is laid surrounding some sort of cytoplasm, and they will form 3 antipodals. 3 antipodals. So here you can count the number of cells and number of nucleus. So 1, 2, 3, 3 antipodal cells, and here 1 egg cell and 2 synergid cells. So 3 cells are also present at the micropylar end. Total 6. And what about this central cell? So total 7 cells are there. Total 7 cells are there. Now count the number of nucleus. 3 antipodal cells, 3 nucleus at the micropylar end, 1 from the egg and 2 from the synergid. To total 6. And the central cell is having the 2 polar nuclei, so total 8 nucleus. So always remember this. The mature female gametophyte or the embryo sac consists of 8 nucleus and 7 cell structure. And this 8 nucleus and 7 cell structure has been formed from 1 megaspore only, and that is why the development of this embryo sac is called as monosporic development. It is called as monosporic development. Okay. Now the nucleus of the functional megaspore divide mitotically to form the 2 nuclei, which move to the opposite poles, forming the 2 nucleate embryo sac. This is talking about that thing which we have discussed. Okay. The nucleus of the functional megaspore divide mitotically to form the 2 nuclei, and that moves to the opposite pole, 1 each. Okay. So this is the 2 nucleate embryo sac. Now 2 more sequential mitotic nuclear divisions. So total how many nuclear mitotic divisions are there? 3 mitotic division, and result in the formation of 4 nucleate and later on 8 nucleate stage of the embryo sac. 8 nucleate stage of the embryo sac at the end of 3 mitotic nuclear division. So this is a free nuclear division. This is a free nuclear division. No cell wall is laid at the simultaneously. The cell wall is not laid simultaneously by the nuclear division. Okay. So it is the interest to note that these mitotic divisions are strictly free nuclear. That is, the nuclear divisions are not followed immediately by the cell wall formation. Cell wall is not formed immediately. Once the 8 nucleate stage is achieved, then only the cell wall is laid. The cell wall is laid and the individual cells are formed. Okay. So 7 celled and 8 nucleus, don't forget. Very important. After the 8 nucleate stage, the cell walls are laid down, leading to the organization of the typical female gametophyte or the embryo sac. Hmm? So, 8 nucleate stage, the cell walls are laid, leading to the organization of a typical female gametophyte or the embryo sac. So, these terms are very, very confusing. Once you read, you understand. But if I ask is after a week, embryo sac, female gametophyte, right? So, just write it at some place that this female gametophyte and embryo sac are same, and in bracket also you must write 8 cell, 8 nucleate and 7 cell structure. So you will remember this female gametophyte, embryo sac, and this embryo sac is 7 cell, 8 nucleate structure is all same. Yes, meiosis. No. You are confusing here. The 4 megaspore, you told 4 megaspore. So 4 megaspore is the reduction division and that is the meiosis. So meiosis is the reduction division. But here, after out of 4 megaspore, 3 get degenerated and 1 is functional. 1 is functional. Now here what I am discussing, the division which leads to the 1 functional megaspore, which is left 1 functional megaspore. So this one is mitosis. This one is mitosis. Okay, simple division. 1 nucleus divide into 2, 1 cell divide into 2. But that in the case of nucellus cell, MMC, microspore mother cell, that undergo reduction division, meiosis, and that form 4 haploid cells, and that is 4 megaspore. Okay, out of those 4 megaspore, 3 get degenerated and 1 is functional. Now in that 1 functional megaspore, this 3 mitotic division takes place and it forms 8 nucleate stage. Clear? There should be no confusion in this. Okay? Okay, great. So here you can see, again, this figure is, because this figure is very important. Yes? So remember this. At the micropylar end, we are having egg and synergid. Egg in the center, synergid side by side. And this structure, the egg and 2 synergid are constitutively are called as egg apparatus. It is called as egg apparatus at the micropylar end. And at the chalazal end, there is antipodals. There is antipodal. We will discuss each and every cell significance. What is the significance of this? Okay. What is the significance of egg? First point. What is the significance of egg? It participate in the fertilization process. Egg participate in the fertilization process. Very good. It forms zygote and zygote leads to the formation of embryo. Good. What is the function of synergid then? Synergid. Synergid contain filiform apparatus and helps to guide the pollen tube to come enter inside the synergid cells and release all the content from the pollen tube. So it guide pollen tube. Very good. This is the function of synergid cell. What is the function of antipodals? Antipodals. Nourishment. Very good. Nourishment. It provide nourishment. It get degenerated and the degenerated cell provide nourishment to the developing embryo. Good. What is the function of central cell? Anyone? Central cell, which contain the polar nuclei, 2 polar nuclei. Central cell. See, after the second fertilization, which is also called as triple fusion. First fertilization is called syngamy and the second fertilization is between the 2 polar nuclei and 1 male nuclei. So that leads the triple fusion. Now this, after the triple fusion, there is 3N nucleus, and that central cell will now called as primary endosperm cell. Primary endosperm cell. Pen. PEN. Pen means primary endosperm nucleus. That nucleus is called primary endosperm nucleus. Okay. Okay. Moving forward. Observe the distribution of cell inside the embryo sac. In the embryo sac, you have to observe what are the different cells. 6 of the 8 nuclei are surrounded by the cell wall and organized into the cells. And the remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. The polar nuclei is situated in the center of the central cell, just below the egg apparatus. Just below the egg apparatus. There is a characteristic distribution of the cell within the embryo sac. Okay, what is that distribution? 3 cells are grouped together at the micropylar and constitute the egg apparatus. At the micropylar, 3 cells are grouped. What are that group? 1 egg and the 2 synergid, and that called as egg apparatus. The egg apparatus in turn consists of the 2 synergid and 1 egg cell. So this we have discussed in details now. Female gametophyte. The synergid have the special cellular thickening at the micropylar tip that is called as filiform apparatus. It is like a finger-like projection. Finger-like projection. Okay? Filiform apparatus, which play an important role in guiding the pollen tube in the synergid. So, if it is asked the filiform apparatus function, you will write it helps in guiding the pollen tube. If it is asked what is the role of synergid, since the filiform apparatus is a part of the synergid, again the function will be the same for the synergid. Okay? And it receives all the content of the pollen tube also. 3 cells are at the chalazal end and are called as antipodals. 3 cells are at the chalazal end. Okay. Apical end. The large central cell, as mentioned earlier, has 2 polar nuclei. Now, this is the central cell. We have already discussed it is having the 2 polar nuclei. And thus the typical angiosperm sac at maturity 8 nucleate and 7 cell structure. So again, this is the 8 nucleate and 7 cell structure, this particular figure. This is the final embryo sac in the angiospermic plant. Now we will talk about the double fertilization. After entering one of the synergid, the pollen tube release the 2 male gamete into the cytoplasm of the synergid. Now this pollen tube, from where it get enter inside the ovule? At which point it get enter inside the ovule? Can anyone tell me? Micropyle pore. Yes, micropyle pore. And micropyle is formed by? It is formed by integuments. Central cell, antipodal cell, and egg and synergid form the 7-celled embryo sac. Yes, 7-celled and 8 nucleated structure. That is the embryo sac. Okay. Now my question was how the micropyle was formed. Micropyle was formed by the integuments. The integuments which covers the embryo sac at one point, they do not join each other and there is a pore-like structure. That pore-like structure is called as micropylar pore, and that end is called as micropylar end. Where this embryo sac is present, where the egg apparatus is present. The egg apparatus is present at the micropylar end. So, from the micropyle pore, this pollen tube get entered and one of the synergid receives the pollen tube release. The pollen tube release, and what it release? It release 2 male gametes into the cytoplasm of the synergid. Okay. One of the male gametes move towards the egg. Now, very important point. Two male gametes were there, and out of those two, one of the male gamete moves towards the egg cell and fuses with its nucleus. Which nucleus? Egg cell nucleus, and completes the syngamy process. First fertilization. First fertilization. Okay. Are the male gamete cells or nuclei? Actually, it is the cell only, but they are having very less amount of cytoplasm. Only small bit of cytoplasm is attached with this male nuclei. So basically it is called as male nuclei. And you will study later on in the human reproduction also that in the fertilization process in human also, only the nucleus of the male is involved in the zygote. The zygote cytoplasm is actually the cytoplasm of the female egg cell, not of the male, because male there only gives the what? Nucleus. Okay. So it's having very less amount of cytoplasm. So that is why it is called as male nuclei or male gamete. Okay. So syngamy takes place by the fusion of one of the male nuclei with the egg nucleus, and that is called as syngamy. Very important. What is the result of syngamy? You all know, zygote, and zygote later on develops into the embryo. This result in the formation of diploid cell that is called as zygote. So here you can see with the help of diagram, clear diagram is there. Filiform apparatus, male gametes are released here. It makes its way, one of the male gamete makes its way towards the egg nucleus, and another moves towards the pole nuclei. Okay. Now the other male gamete moves toward the two polar nuclei located at the central cell and fuse with them to produce triploid primary endosperm nucleus, PEN, primary endosperm nucleus. Triple fusion. This is called as triple fusion. And two times the male gamete and the female gamete fuse together. Two times. Okay. And that is why the syngamy and triple fusion together, it is called as double fertilization. It is called as double fertilization, and this is the very unique feature of the flowering plants. Very unique feature of the flowering plants. So here you can see primary endosperm nucleus, PEN, and this central cell, here you can see primary endosperm cell, central cell name got changed. As the triple fusion takes place, the central cell, earlier it was this particular cell, it was called as central cell, but once the triple fusion takes place and the primary endosperm nucleus formation takes place, this central cell is now called as primary endosperm cell. This is called as primary endosperm cells. Clear? And here you can see the zygote is there. Now the degenerating synergid. Synergid has done its function. It has done its function. What is the function of central cell? What is written here? You will answer yourself. Primary endosperm cell. Endosperm. So this primary endosperm cell was actually earlier central cell, and this will later on form the endosperm. Now this zygote will form the embryo. This zygote will form the embryo. This will form the embryo. And this primary endosperm cell will form the endosperm. Endosperm is important because the reserve food material comes from this endosperm only, and it provides to the developing embryo. It provides to the developing embryo. So this primary endosperm cell is nothing. Earlier, just before the triple fusion, it was central cell, but after the triple fusion, it is called as primary endosperm cell, and the nucleus is called as primary endosperm nucleus. Okay, good. So as this involved the fusion of the three haploid nuclei, it is called as triple fusion. Since the two types of fusion, syngamy and the triple fusion, take place in the embryo sac, the phenomena is termed as double fertilization, an event unique to the flowering plant. This you see only in the flowering plant. The central cell, see this point. The central cell after triple fusion becomes primary endosperm cell and develops into the endosperm, while the zygote develops to the embryo. Now, which will form? Embryo will form earlier or endosperm will form earlier? Which will form early? An embryo or endosperm? Which will form early? First embryo or endosperm? Okay, I got two answers, endosperm and embryo. So, first of all, endosperm will form and later on embryo will form. You can ask why? Because the formation of embryo required nourishment. And when the endosperm will form, the nourishment will come from the endosperm. Yes, as embryo need food, embryo need nourishment. So from where this nourishment is coming? It is coming from the endosperm. Though the zygote forms earlier and triple fusion takes place later on, but still the endosperm will form, the embryo will remain at that particular stage only, and when the endosperm will form, then this zygote will start dividing and will form the embryo later on. Okay. Summary: in seed plants, the male gametophyte reduced to the pollen grain carries sperm cells and the female gametophyte embryo sac within the ovule. We have discussed this. The female gametophyte develops within the ovule and contains the egg cell. Double fertilization is unique to the flowering plants, where one sperm fertilizes the egg and forming a zygote diploid. That is the syngamy. Result of the syngamy is zygote. And while other fuses with the polar nuclei, giving rise to the endosperm, which is a triploid structure and which nourishes the developing embryo. This process ensures the seed contain both the embryo and its food source. The seed contain both. The resulting seeds then develops into the new sporophyte generation. So, this is all about today's session. Now, quickly we will have our DPP. Just give me 1 second and I'll come back with the DPP. Okay, so first question is here. Okay, one of the student asked some question. I will answer you after this DPP. Which of the following is the result of the syngamy in double fertilization? Which of the following is the result of syngamy? Let us see the option here. Formation of primary endosperm nucleus. Second, formation of zygote. Third, formation of triploid cell, and D, formation of endosperm. What is the result of the syngamy in double fertilization? So you all know better. Okay, formation of zygote, formation of primary endosperm nucleus, that is result of the triple fusion, right? So this could not be the answer. Formation of triploid cell, again that is the central cell only. And formation of endosperm, it is again talking about the triple fusion only. But the formation of zygote is the result of the syngamy, and that is the part of the double fertilization. So I'll opt the second option and save and move to the next question. The next question is here. What is the ploidy level of the primary endosperm nucleus in the flowering plant? Ploidy level of PEN, primary endosperm nucleus. It is haploid N, it is diploid 2N, it is triploid 3N, it is tetraploid 4N. So, which option is correct? Triploid 3N. Yes, primary endosperm nucleus. Two polar nuclei and one male gamete fuses together, and that fusion is called as triple fusion. The nucleus is now called primary endosperm nucleus, because by the division of this nucleus only the endosperm will be formed. So that is why it is called as primary endosperm nucleus. Okay, great. We will save this answer and move to our next question. Next question is here. Double fertilization in the flowering plants involves, first, A, fusion of the two male gamete with one egg. Second, fusion of one male egg with the egg and another with the polar nuclei. Third, fusion of two male gamete with the two polar nuclei. And fourth, fusion of one male gamete with the two polar nuclei and another with the synergid. So, which is the correct answer? First one, fusion of two male gamete with one egg. It never happened. Two male gamete with one egg. No. So, first one is incorrect. Fusion of one male gamete with the egg. Yes, this part is correct. And the another with the polar nuclei. This is also correct. So, the second option is correct. But let us see the third and fourth also. Fusion of the two male gamete with the two polar nuclei. This is incorrect. Fusion of one male gamete with the two polar nuclei and another with the synergid. Synergid fusion with the synergid, male nuclei never fuses together. So this is again incorrect. So option 2 is correct. We'll save it and move to our next question. Which structure develops into the endosperm after double fertilization? Which structure develops into the endosperm after double fertilization? Let us see the option. A, zygote. B, primary endosperm nucleus. C, synergid. And D, egg cell. Which structure develops into the endosperm? Second, PEN, primary endosperm nucleus. Very good. So we will opt this as our correct answer and we will save and move to our next question. What is unique about double fertilization in flowering plant? Unique feature they are asking. A, formation of two zygote. B, involvement of three male gametes. C, fusion of three haploid nuclei in a triple fusion. And D, occurrence of meiosis after fertilization. So, which is the correct answer? Unique about the double fertilization is flowering plant. Fusion of three haploid nuclei in the triple fusion. This is the unique feature of the flowering plant as called as double fertilization, and it is a part of the double fertilization. So we will opt option 3 and save it and submit our test. Let us see the result. Okay, so all answers are correct here. Okay, so this is all about today's session. We learned today about the double fertilization. So go and revise this chapter once again, and we will meet tomorrow with the new topic. So thank you very much for attending today's session. Take care, and you will have your another class. So please join this class, that class. Thank you. ये हो गया है मेरा यहाँ पे